The overall goal of this procedure is to investigate the dynamics of long, non-coding Xist RNA and its associated epigenetic modification during X-chromosome inactivation by using a quick fluorescent in situ hybridization combined with immunofluorescence. This protocol requires cytospin preparation of slides with differentiating embryoid body cells, which are permeabilized and then fixed for staining. Next, fluorescent marker localized by an antibody to histone H3 trimethyllysine 27 accumulates on the inactive X chromosome. The final step uses oligonucleotide probes that contain a 5' amino modification and are labeled with an amine-reactive fluorophore to perform Xist RNA FISH. Ultimately, fluorescence microscopy is used to show the localization of Xist RNA, cloud, and histone modification during ES cells differentiation.
The main advantage of this technique over existing methods is that we use multiple unique oligonucleotides which are labeled with a single fluorophore. This provides us with strand-specific detection, which reduces the background, and this also increases the penetration into the nucleus, and it increases or enhances the intensity of the signals. In addition, when we increase the concentration of the probes, we can reduce significantly the time required for completing this immunofish experiment.
Though this method can provide insight into the localization of Xist RNA on the